Right, hello everyone. It's our live talk week this week at Live with Scientific. Thank you again for joining us for this live talk with Dr. Miriam Smith on nervous system tumours. And our genetics month is going to continue for another until the end of this month. And we have got some hands-on activities which are prepared by our volunteers. So please make sure that you're tuned in to find out about the other material that we have prepared for you. So again, for those who are who just joined us this week, my name is Aisha and I'm directing the Live with Scientists project with the help of so many people. And all in all, what we're trying to do is we're trying to communicate our science to everyone, regardless of time and space. So this is the reason why we have started to actually live stream and archive our talks online. So you can, if you or anyone in future, if you're missing any of these talks, please feel free to actually join our Facebook group or to join to our other social media accounts, or to watch the videos that we have on YouTube that are archived on YouTube as well. So I'm so pleased to have Dr. Miriam Smith with us today. So Miriam is an expert on schwannomatosis and neurofibromatosis that we see in brain. And I'm sure these are really mouthful and hard to remember. And I'm sure she will be explaining a lot about these in her talk in a minute. But before I actually leave the stage to her, firstly, I would like to introduce her to you. So Miriam has actually completed her studies in UK and then moved to Boston for her postgraduate postdoctoral work. And after completing that work, she then brought all of that knowledge and experience back to UK, to University of Manchester, and has been doing really significant work since then. So Miriam, thank you very much for your time today, and I'm really looking forward to listening to the recent advancements that you had in your field and in your research, and the stage is yours.
Okay, thanks very much. Thanks for inviting me to speak. I'll just see if I can share my slides. Okay, can you see that?
Yes, I think we can.
Yeah. Okay. So, yes, thanks again to Aisha for inviting me. I'm going to talk to you today about some of my work on nervous system tumours and the diagnostic testing that goes on with that. So I'll just quickly go through a summary of my talk, if I can get my slides to move. Here we go. Yes, so the talk structure, I'll start with a little bit of background about me and the journey to where how I got to where I am now. I'm going to do a bit of an introduction to nerve sheath tumours, which is the main thing I'm going to be talking about today, and some of the gene changes that are known to occur in these tumours, before going on to schwannomatosis and the genetics of schwannomatosis, and some of the genetic testing strategies that are used in the clinic to identify whether or not someone has a predisposition to schwannomatosis, and then a specific case study, which is one of the most recent things that I've been doing in the lab, which is one of the ways of looking at how to identify some of the more unusual gene changes that are not really picked up on routine genetic testing strategies that are used every day in the clinic. So some of my background, just to start from the beginning, I grew up, went to school mainly in London and in Liverpool. So having moved from London to Liverpool, I decided to go off to university, to Bath University, and there I was studying molecular and cellular biology, which is this degree here, and that included a placement. So that's what's known as a sandwich degree. So that had a 1-year placement after year 3, or after year 2, and then that was year 3, and then I came back for year 4. So I ended up going a bit backwards and forwards between Bath and Liverpool. There was a 1-year placement in industry in a pharmaceuticals company where they develop vaccines. And then went back for my final year to get this degree. And the main thing that I really enjoyed doing during that degree was looking at neuroscience. So I decided to do a PhD in molecular neuroscience, which I did in London. That was at the UCL School of Pharmacy. So I got my PhD, and then I stayed there for another year to do a little bit more research in the field that I was working on, which was at the time in neurotransmitter receptors and how they're assembled in cells and how they work. And then after that, I decided I really wanted to have more of a clinical aspect to my work, and I wanted it to be a bit more applied, that it was a bit more obvious why I was doing these things and what the benefits were. So I ended up leaving London and went off the map and ended up in Harvard Medical School. And I was there for about 4 years, initially at the Brigham and Women's Hospital. So I arrived there in 2005, and I started working on genetic models of Alzheimer's disease. And the link there was with the neurotransmitter receptors. And then in 2007, there was a major development in the field of schwannoma and schwannomatosis genetics. And I ended up moving to Massachusetts General Hospital to start working on genetics of schwannomas. And then in 2009, I decided I wanted to continue this work. And when I came back to the UK, I ended up in the University of Manchester. And so I've been there since 2009, and I wanted to continue working in the similar field. So I applied for some money to support my research. And in 2011, I got a Young Investigator Award from the Children's Tumour Foundation. And that was really to look at some more gene discovery for the underlying causes in schwannomas and meningiomas. So there are two different kinds of tumours that you find within the nervous system, and I'll be talking about them more. And the main thing that came out of my study with this award was that we discovered a novel tumour predisposition syndrome that was very specific and predisposed to multiple meningiomas. And we identified this by discovering the gene that causes them, which is the SMARCA1 gene. So having published this paper, I decided to stay at the University of Manchester, and I became a lecturer in 2013 in cancer genomics. And then just this year, I've become a senior lecturer in cancer genomics. So that's how I came to be doing what I'm doing now, setting up my own research group with PhD students and postdocs and continuing this work. So the main thing I'm going to be talking about today is nerve sheath tumours. And this is just a slide to give you a bit of background of nerve sheath tumours. So the tumours that I'm interested in are schwannomas, which are benign tumours, and they're derived from Schwann cells, which is how they get their name. And these Schwann cells, if you can look at this diagram here, this yellow bit here represents a nerve. So this is the axon of a nerve cell. And then the blue bit, sort of bluish bit surrounding it, this is a Schwann cell with the nucleus here. And this cell wraps itself around the axon. You can see that it's getting more and more layers here. And these cells act like an insulator. So much like an electrical cable, the axon is sending electrical signals throughout the body. And the Schwann cells act like an insulator to protect the nerve. And if you have a tumour, they can end up looking like this diagram here in the black box, where the cells have overgrown. And these kind of tumours can occur anywhere throughout the body on the nerves. They account for just over 10% of all brain and nervous system tumours, and the vast majority of them are vestibular schwannomas. And this red arrow here gives you an approximate indication of where the vestibular nerve is. So it's in the brain, and it's the most likely place that a schwannoma will grow. So looking at some of the causative gene changes that you might find in schwannoma tumours, the most common finding is a variation in the Neurofibromatosis type 2 gene. Now this is, I'll give you a little overview of where the gene is. You can see this box in blue here, which gives you a normal human karyotype. So this is your complete set of genes organized into chromosomes, and you have 2 copies of each set. So you have 23 different chromosomes, and you have 2 copies of each one. one set that's inherited from your mother and one set that's inherited from your father. So the NF2 gene is located on chromosome 22, that's the little one. And if I've got this enlarged version of chromosome 22, which indicates the location of the NF2 gene. So if we look at this box here, which would represent the chromosomes that you'd find in an individual cell, and this would represent a Schwann cell. You have two chromosome 22s there. If you acquire a mutation in one of your copies of NF2, you have one damaged version, but you still have one normal version. And that normal version is able to compensate for the lack of function in the damaged NF2 gene. However, if you acquire a second variant, which occurs on the other allele, your other chromosome 22, you then knocked out both copies of NF2, and that's then not functional anymore. And that's what would happen inside a Schwann cell when a Schwann cell turns into a schwannoma tumour. That will start to overgrow and develop too many cells, and then you end up with a tumour. So there's a particular disease called neurofibromatosis type 2 disease. And in that scenario, you've ended up with a variation, a damaging variation in one of your copies of NF2 in every cell. So the prevalence of this disease, it's relatively uncommon. You have, within the UK, 1 in about 67.7 thousand people. And the reason why you'd have one of these variants in every cell is that it's been inherited from a parent cell. So it's either in the egg or the sperm from one of your parents. And so because of that, you've ended up with a predisposition that means that you only then have to inherit one more variant before you end up developing a tumour. So this blue box here would represent what you can find in every cell in the body, not just Schwann cells, but every cell in a person who has neurofibromatosis type 2 disease. And if they then develop a single extra variant on the other copy, they have then developed a tumour. So it makes them much more likely to develop one of these tumours. And within NF2, schwannomas are the most likely tumour that they will develop, but they can also develop meningiomas, which occur in the meninges that surround the brain and spine, and they act as a barrier for preventing bad things from getting into the brain and spine. And ependymomas, which in NF2 are mainly found in the spine. And again, in NF2, vestibular schwannomas are the most common type of schwannoma that you'll find. And the hallmark of NF2 is that people with NF2 disease will develop bilateral, i.e. 2 vestibular schwannomas, one on each side, so that these nerves, the vestibular nerve, affects hearing. So if you have large tumours on both sides, one of the main effects is that you can become completely deaf. It also causes problems with balance, and with any of these tumours, they can lead to more severe consequences if they're large or if they impinge on something important within the brain or spine. And this is just to show you where the vestibular nerve is. So this is the brainstem, this yellow bit in the middle here. Then there's the outline of the brain. This is where your ear is, and then the nerve goes from your ear to the brain, and the brown blob there is a vestibular schwannoma forming. So another condition which also predisposes to schwannomas is called schwannomatosis. And this is less common than NF2, and the prevalence in the UK is around 1 in 58,000. But in this condition, you tend to develop non-vestibular schwannomas. They tend to be more on the peripheral nerves. So this diagram at the side shows you an MRI scan, an imaging scan of a whole human body of someone who has schwannomatosis, and the white arrows are indicating some of their tumours. So you can see they can be all over the body. And they tend to be non-vestibular, so it is different from NF2. And also they're more likely to suffer from severe pain, possibly because their tumours are more likely to occur in the periphery. So there are 2 genes that are known to cause schwannomatosis, and they are the SMARCB1 gene and the LZTR1 gene. So the SMARCB1 gene was discovered in 2007 to be associated with schwannomatosis, and that was the major event that I mentioned briefly about why I ended up working in this field. So because this gene was discovered, schwannomatosis was distinguished from NF2, and it became a whole extra field of research to work in. And then in 2014, the LZTR1 gene was also associated with schwannomatosis. So a confusing thing about these genes is that if you have LZTR1 associated schwannomatosis, around 5% of people with this kind of schwannomatosis will develop a vestibular schwannoma, and it's usually unilateral. But it can confuse the diagnostic criteria. It can be a bit confusing to know which disease they have. And then people with SMARCB1 associated schwannomatosis, around 5% of those people will develop meningiomas. So again, there's some overlap with NF2. So to look at these genes that are involved in schwannomatosis, you'll notice that they're all found on the same chromosome. So NF2, which was discovered first, is the furthest away from the left-hand side. Then SMARCB1 is in the middle and LZTR1 at the far side, and they're all within 6 million bases of each other. So it sounds relatively far away, but remember, this is one of the smallest chromosomes. And that's actually very interesting and complicating for the genetics. So what happens in schwannomatosis is, this is again representing a cell, a Schwann cell. And I've started with a SMARCB1 variant, but this in schwannomatosis could also be an LZTR1 variant. So this is your first hit. This is your germline mutation that's found in all of your cells. And then you end up with your second hit, which knocks out the other allele in the same mechanism that you would have for NF2. But what you also find in schwannomatosis is that you tend to find, if you look in a tumour cell, if you look at the DNA in a tumour cell, that there's this extra hit in NF2. So you still, within the tumour cell, have loss of both copies of NF2. So you end up with a greater number of variants in schwannomatosis. And this is another reason why it's been traditionally very difficult to distinguish between them. Because if you're just doing tumour analysis and you find NF2 variants, you might think they have NF2 disease. But then if you look in their blood, in some tissue or form of DNA that doesn't come from the particular tumour, you don't see that variant and you're wondering why it's not there. It may be that they actually have schwannomatosis. So in terms of predisposition, LZTR1 and SMARCB1 variants can be found in around half of all people who have schwannomatosis. And SMARCB1 variants, which are the main type of variant I'm going to be talking about today, are found in around half of people with schwannomatosis that is found in families, and in around 10% of people where it's just an individual case where they're not aware of being related to anyone who has the disorder. And this diagram at the bottom shows you the chromosomal locations of LZTR1, SMARCB1 and NF2. And then underneath, I've expanded just this region of SMARCB1. So this is the gene structure of SMARCB1. And it has, I'll go into this in a bit more detail, but it has 9 exons, and they're spaced with these intervening intronic regions. So just to take a brief interlude to explain something about gene processing for people who are not familiar with that, is that you may know that the DNA has 2 strands. You have your double helix of DNA. That's then organized into these coding regions, the exons, and the non-coding regions, which are these intronic spaces in between. And they tend to have regulatory elements in them. This DNA sequence is then transcribed into RNA, which is represented by this red version of SMARCB1, and this is single-stranded. So RNA is the single-stranded version. It then goes through a series of processing stages called splicing, where you end up chopping out, you snip out the intronic regions and piece all of the coding exonic regions together. And this gives you your RNA transcript, which you can see at the bottom. So after this stage, this is the RNA transcript. It goes through translation to turn it into a protein. So in the middle here, this big pink circle represents a ribosome, the big translational machinery that helps you to process the RNA to form proteins. So this is the single-stranded RNA sequence. And these individual tRNA molecules bring in an individual amino acid that's going to link together to form your protein sequence. So the individual letters at the top represent the amino acids which are forming the protein. And then that protein falls together to produce the final protein structure. And this here is the final structure of SMARTB1. And if you can see the dotted lines, that indicates how much of this protein is represented by each individual exon from the RNA. So in terms of the type of variants that you would find in SMARTB1 that cause schwannomatosis, the very first variant that was identified, which was enabled SMARTB1 to be linked to schwannomatosis in the first place, was called a nonsense mutation. So it had a nucleotide change in the DNA sequence, which led to a stop in the protein sequence. The blue box here represents the DNA sequence and the protein sequence. And you can see highlighted in red where there's been a spelling mistake, which has meant that it's changed what would be represented in the protein at that position. And what actually happens is that it causes this stop to appear, and then the rest of the protein is not produced. So what normally happens with these kind of nonsense mutations is that the protein is degraded, it's never produced, and you end up with no protein at all. Another type of variant, which is much more common in schwannomatosis, is a missense mutation, where you can see highlighted in red here, there's been another spelling mistake, but rather than preventing the protein from being produced, it's just changed the spelling. So the proline there has been turned into a histidine. Another type of variant which is common in schwannomatosis is a splice site variant. And you can see in the sequence in the blue box here that there's a G that's been highlighted, and that got changed. And you can see here also that the DNA lettering is in capital letters up to the G, and then it's in lowercase letters after that. So that's where the end of the exon is, and the lowercase letters are in the intron, which is not used to produce protein, but it has regulatory regions in it. And if you have a variant that occurs around these junctions, that tends to cause problems with splicing. So when you're snipping out pieces, that kind of the machinery that regulates that can go wrong. And then the last type of variant is within a regulatory region. It's not actually in an intron, it's after the end of the coding sequence. So the normal wild type stop codon, which is represented by this black asterisk here, is followed by a series of these lowercase letters, which is not in a coding sequence. And then in this particular case, 82 nucleotides after the end of the stop codon, there's a change in the sequence. And it's not 100% clear what this is doing to the protein, but it is the most common variant that you'd find in schwannomatosis. The greatest number of people who have schwannomatosis have this same variant. So this is just a diagram to show you some of the specific variants that have been found in people who have schwannomatosis using genetic testing in the clinic. And I've highlighted some in green, which represent splicing variants, and some in red that represent missense variants. The ones that are highlighted in with a green background, you'll probably notice they look like they should have been nonsense mutations and they've made a stop, or they should be a missense mutation. But when I've actually looked at some of these more closely and done some research to investigate their mechanism of action, they've actually turned out to be splicing variants. So sometimes it can be confusing, even if you think you know what kind of variant you have, to know whether that's really how it's behaving. And the ones that are highlighted in yellow are the other variants that are supposedly nonsense mutations. But what actually happens in these cases is that the group that actually identified the original variant in schwannomatosis, the original nonsense mutation, did a series of studies of these very early, very 5' at the beginning of the gene, these variants that occur there, which look like they should be leading to a complete loss of protein. And in fact, what's happening is that they are reinitiating the start of the protein coding region a little bit later on, and it's close enough to the beginning of the gene that it's been able to produce most of the protein, but just with a little piece missing at the beginning. So it's actually ended up behaving a bit more like a splicing variant or a missense variant. You end up with protein, but just maybe not quite as not functioning quite as well as it would be if you had the entire protein sequence. So now just to go through some of the genetic testing strategies that are used in the clinic routinely to identify some of these variants. This is, again, a map of SmartB1 at the top, and these arrows indicate where primers are located to decide where you might want to investigate. And because we know that most of the pathogenic disease-causing variants that are found in schwannomatosis are found in these coding regions, we want to amplify those regions and have a look at them in more detail. So we map out with these little primer sequences the bits that we want to amplify. They're then put into a plate with enzymes and with the DNA template, which is DNA that's been extracted from cells from a patient who has schwannomatosis. That then goes into this PCR machine, which goes through a series of thermocycling heat changes to go hotter and colder, depending on whether it's amplifying the DNA, whether it's at the optimum temperature for different bits of the enzyme activity to work, or to break the DNA apart so that the enzymes and the primers can get to where they need to go to amplify these regions. And this will end up making lots and lots of copies of the template DNA. So you end up with a large number of them, so that when you try and have a look at them, you're judging from a large pool of sequences. You can then, when you've run them in the PCR machine, you can run them out on an agarose gel to see whether your reactions have worked. So each of the lanes on this gel here represent a fragment of DNA that's been amplified. So there's a DNA ladder at the beginning, which will indicate various different sizes of DNA, so you can tell whether what size your fragment is. And then each of these rows here represents several different patients' samples of the same region, and they've all been amplified. These are then sequenced, so they are, this bottom line here represents the template DNA sequence. Then you've got your primer sequence, which binds to the beginning of the sequence, so you know where you want to start. And then you put this in with a pool of different nucleotides, which have all been broken down into the individual nucleotides, and they've been labeled with different colors, so that at each position, so on this bottom row, this will be in a particular sequence. Each nucleotide will be in a particular order, and whichever base or nucleotide is at that position, a fluorescently tagged version of that will join this top row until you end up with, you're building up this sequence, and each of these letters will be tagged with a different color depending on which letter they are. And you can then sequence this to see what's in there. So when you're looking for single nucleotide changes in the DNA, there are 3 possible combinations of what you might see at any given location. So in these diagrams here, we have the 2 alleles. So remember that when you amplify these from someone's DNA, you're amplifying 2 pools, the version they've inherited from their mother and the version they've inherited from their father. And at each position, you can have, in this case, both of them. In this case, they've got 2 different copies. So one of the parents has passed on a T at this position, and one parent has passed on a C. And then in this version, both parents have passed on a C. So you can look at a particular SNP location and see what is their genotype, what is their individual genotype at that location. When we then look at next generation sequencing, it does something similar, but on a much larger scale, and we tend to be able to sequence longer regions. So the exons are maybe 1 to 200 bases in size. And when you're looking at next generation sequencing, if I do this, you can see that the beginning and end of where you want to amplify is expanded. So you're looking at a much larger region. And this is a diagram that's been taken from the NOMAD database, which is a freely available online database. And this is a map of the coverage, the sequencing coverage of the SMARTB1 gene. So you can see that there are peaks at each of the exons, and the blue peaks indicate the exomes. This is just the coding regions. And then the green is indicating the genomic sequence, so it's more uniform throughout the whole gene. And in the panel sequencing that's used for SMARTB1, it would look more like these blue peaks here. So it would be the regions in and around the exonic areas, and they will be covered to a very high level. Another thing that we might use is copy number analysis to see whether there is a whole gene has been deleted, or whether a single exon has been deleted. This is an example of the NF2 gene. So each of these black lines at the top here represents a probe which is detecting whether or not the exon is present. And this diagram here shows the light grey indicates a normal copy number of 1. So this is what you would normally expect to find in a cell. And that represents it actually represents both copies, so 2 copies, but that's counted as 1. If you lose some of these markers, then these are indicated in the black. This represents a whole gene deletion, where you can see that any of the control probes are the same as the normal wild type controls. And then the black ones, which are in exons, have been deleted, and they're represented much lower. So that indicates that there's been a large change there. Another thing that they might use in the clinic is microsatellite analysis to look for loss of heterozygosity, which will tell you whether or not you have 2 copies or 1 copy, but it won't necessarily tell you about the copy number. It will tell you whether they are the same or different. So this indicates here, this is a region of chromosome 22, and the bit that's been highlighted in bold and underlined, you'll see that that's very repetitive. So it's a repeat region. And what you might expect to find is that somebody might have different numbers of copies of that region. So that fragment will be bigger or smaller in different people, and possibly on different chromosomes within the same person. So this is just an example to show you that in blood, this is someone who has 2 copies of different sizes. This one is 160 bases big, and this one's 167 bases big. Whereas if they look in the tumour, this smaller copy is here, and the larger one has disappeared. So that's an indication that you've lost some genetic material, or that only 1 copy is represented in the tumour. So the last part of the talk is, the main bit is to go through a particular case and indicate the type of clinical testing they underwent, and what happens when you can't find something using these routine tests. So the particular case is this large schwannomatosis family. So this is their pedigree, and you can see that all of the people who are indicated with a black circle or a black square have schwannomatosis. And then this person with a white circle does not have schwannomatosis. And within this family, so there are multiple people who are affected. There's a lot of variability in the severity of the disease in this family. Some people have a lot of schwannomas that cause severe pain, and they'll have a lot of brain tumours and peripheral tumours. And because they have such a wide variety, such a variability, and some of those phenotypes are very severe, there have been a lot of requests within this family for people to be tested to find out what's causing it, and they want to know a specific gene change so that if they want to have a baby, they can tell before it's born with pre-implantation genetic diagnosis or prenatal genetic diagnosis whether or not that baby will be affected, because they don't want to pass this on. So they were tested because they had such variability in different symptoms. It was difficult to tell whether they would have NF2 or schwannomatosis. And they have, over the years, been tested using Sanger sequencing, which was the original testing that I showed you, the chromatograms from, the coloured charts. And then they also underwent next generation sequencing with the wider sequencing range. And they've looked at NF2 and SMARTB1 and LZTR1 and couldn't find any variants in any of these genes. So that makes it more difficult to determine what's causing it and how they can tell before a baby is born whether or not they're likely to be affected. So if they have a look at the tumours in this family, it's one way of looking at, if you can't see something in your germline DNA in all of your cells, is there something specific in the tumours that might give you an indication? And when they've had a look in the clinic at the tumour DNA from these patients, they have, in fact, they looked at 2 different tumours, and these tumours are from 2 different people. And if they look at the DNA in these 2 different tumours, you can see that there's an NF2 variant and loss of heterozygosity in both of the tumours, but the point variant in NF2 is different in each of these tumours. So it tells you that their disease is linked to chromosome 22, but that it's not caused by either of these specific variants in NF2. And you know from earlier that NF2 somatic variants can also be found in schwannomatosis patients. So it doesn't really distinguish between these 2 diseases, but it suggests that it might not be NF2. When they've then had a look at germline DNA, again, going back to their blood DNA, and had a look with some microsatellite analysis to see whether they can determine who has how many copies of which of these microsatellite markers, and whether that can give you any indication of where their variant might be, which gene is causing this. You can see that I've put in squares these regions which are the same in all of the people who have schwannomatosis, but different in the person who doesn't have schwannomatosis. And you can see, for example, in 2 people here, they have the same allele all the way down the chromosome, but these 2 people who are also affected have a different version. So you can localize this down to this region of this 321 here, and that the gene that surrounds is SMARTB1. So it gives you a strong indication that there's something going wrong with SMARTB1, but that we haven't been able to find it looking at routine genetic testing. So this is where some of the more recent research has come into it. They've actually done some RNA analysis, not knowing whether there's a variant in here or not. We've done some sequencing of the full-length RNA transcript. And if we do Sanger sequencing from this position here, so we've actually sequenced the whole thing, but this is the most interesting bit. This is what this region looks like in a control sample. So you can see the actual DNA, or RNA in this case, sequence at this position. If we look at the RNA from somebody from this schwannomatosis family, you can try and spot the difference here. You can see that there is this second sequence running along the bottom here. And if I point this arrow here, this position is the end of an exon junction. So I don't know if you can see in this orange region here, this shows you where exon 6 ends and exon 7 begins. And you can see that from this exon junction, something has gone wrong here. So what has that actually done to the RNA? Well, this is a map of the wild-type RNA with each of the exons labelled, and the dotted lines indicate the exon junctions. And what's actually happened here is that there's been an insertion of some extra sequence between exons 6 and 7. And the asterisk will tell you that it's created a stop here. So what happens is that you get your first 6 exons, then this extra bit of inserted sequence which is not divisible by 3. And importantly, it needs to be divisible by 3 to keep it in frame so that the protein will keep adding amino acids in the right order, because every 3 bases codes for 1 amino acid. So it's actually introduced a stop. And don't be too scared about this diagram. You're not supposed to be able to read all of the text here, but this is just to show you the absolute genomic sequence between exon 6 and exon 7 in the SMARTB1 gene. So this is genomic DNA. The orange bits are exon 6 and exon 7. And this green bit that I've highlighted in the middle in one of the introns, which is normally spliced out, matches the sequence that's been inserted between those 2 sequences. So it's actually mistakenly added a bit of an intron where it shouldn't be. So if we then sequence in the genomic DNA in someone who is affected in this family and someone who is not affected in this family with schwannomatosis, you can see that there are 2 different nucleotides at this position, and where there's only 1 in the person who doesn't have schwannomatosis. And what this does is, this green bit is the same green bit that's been highlighted in the previous slide. This region, this regulatory region around the outside, which regulates splicing, there's been a change here. And this, while it wouldn't normally be a splice site, the base or the nucleotide that this C has changed to, when it changes to a T, that turns it into a sequence which is recognized by the splicing machinery as a splice junction. So it's created a splicing junction where there normally wasn't one, and it's been inserted incorrectly into the RNA. So if we have a look at the bottom again of the map of the SMARTB1 gene, this arrow indicates where this one nucleotide change has occurred. And if we think, why wasn't that picked up on the next generation sequencing panel? Because that covers quite a large region outside of each exon, maybe up to 1,000 or 1,200 bases outside of that region. You can see that it's very slightly outside of the coverage. So this would never be picked up on routine testing in the clinic. So this makes it difficult to detect this kind of variant using clinical techniques that are used routinely in the clinic at the moment. But with this kind of research, we can show what's wrong with this DNA, what it's doing, and it can inform how people are tested in the future. So it may be that they want to expand this to cover these regions when we know that there is something in there that can be causing damage to a particular family and causing them to have this disease. And it also means that it can then be used to test people, so that if this particular family then wants to have another baby, someone in this family wants to have a baby, and they want to know, do they have this particular variant? It's much easier to sequence one little region than to go through all the effort of doing the microsatellite analysis and looking at the region and not being completely sure. Like, you're assuming that you're looking in the right region, but you don't have definitive proof. And this kind of thing can give you the definitive proof that you need to go back and test people. So just to summarize what I've shown you, I've given you an overview of nerve tumors and some of the known causative genes. I've shown you that there are still some people who have schwannomatosis in whom we can't find a genetic cause. But that some of these variants will be found in genes that are already known and already associated with schwannomatosis. Although there may be some other variants that are found in genes that haven't been discovered yet. And it's very useful to be able to discover these variants so that you can inform clinical management, allow for prenatal genetic testing, and also to improve our understanding of how these diseases work and what's causing these tumors to grow. That also helps us to develop future treatments, maybe by gene therapy or maybe by how to target new drugs. So that's the end of the talk. I'd just like to acknowledge some of the people who were involved in the study, especially Gareth Evans, who is the clinician who sees a lot of these patients, and the genetic testing team within St Mary's Hospital, and some of the clinicians from Australia who are involved in looking after the family And also Katie and Christina, whose trailers you might have been seeing, they've been working with Live with Scientists. So they're working in my group on the same kind of tumours. So I'm hoping they'll have some similar discoveries coming up soon. And thank you again, Aisha, for inviting me.
Thank you, Miriam, for this informative talk. Now we're going to give a 10-minute break, and once we resume, we're going to start with audience questions, if that's all right with you. It was a very interesting talk, and I'm sure it will actually grab some attention and make people, ask questions about it. So make sure you have a good rest. Hi, Miriam. Hi.
Hi.
Right, I hope your break was nice, and everyone who has been watching us and waiting for us, thanks very much for being around. So, Miriam, the talk was really very interesting, and I think it is really good that there is work that is being done in this field. And I would like to kickstart with a question that I have, if that's okay. So my first question is, just to give our audience a feel of why this is important, can you tell us how this or any other similar studies that you have done is going to help firstly the patients, the field, and other scientists who are actually working in the field?
Yes, well, so with the particular study I was talking about, the main thing that we're interested in is genetic diagnosis. So it's being able to determine what is causing someone's disease, what gene is involved, what specific variants, and then also how does it work? Is it, like I was saying, loss of protein, change of function of the protein, or misregulation of the protein? And then that gives you more information about how you might go about treating someone. So you might be able to, you might find out what cellular pathways the protein is in and what they're doing and how you could target that with a specific drug. And also, I guess also following on from the genetic diagnosis, there is also what I was saying about the prenatal diagnosis and pre-implantation genetic diagnosis. If you've got a disease that's very severe and you don't want to risk passing that on because you don't want a child to have to live with the same condition, you can find out before they're born or before an egg and a sperm are even combined whether or not they're likely to be affected. And then you can choose to select an egg or a sperm that's unaffected to make sure that they're going to be a healthy child.
Great. Thank you very much. Great. Thank you very much, Miriam. So, so the variants that that you identify, you find, how do you know whether or not they actually have a function? Because we know that we have learned from Elke's blog and we have learned from any other materials that we have released this month that, and from Dave Gerard's talk as well, that there are parts of the genome, parts of the DNA that doesn't actually have a known function. So when you identify, or even on a gene as well, there are areas that might or might not be significant for the function or physiology of that cell, of that person. So how do you identify whether or not these variants do actually have a function? What kind of methods do you use for that?
So one thing is that, especially within schwannomatosis, that can be very challenging. What you would normally do in terms of identifying pathogenic variants is look for something that ought to have an effect on the protein. So where I showed the nonsense variants, they look like something that would completely destroy the protein, that you shouldn't have any protein left. So it looks like it ought to have a change. It ought to do something to affect the protein. If you have something that's more like a missense variant, it can be more difficult to tell whether that's something that is normal variation and the protein can tolerate that kind of change, or whether it's going to have a damaging effect. One thing you can do to judge whether or not it's likely to be pathogenic or disease-causing is to look on these public databases like Nomad and Exact and see if you look in a large number of genomes or exomes from people who are not known to have the particular disease you're interested in, do you see these variants occurring in everyone, in a lot of people? And if they're occurring commonly, then it means that the variant is less likely to be pathogenic, because so we know NF2 and schwannomatosis are relatively uncommon. If you see a variant that's occurring commonly, more commonly than the disease is known to occur, it's unlikely to be pathogenic. So one thing is to look at frequency, how commonly you find these variants. If they're extremely rare, then they look like they're having an effect on the protein, it gives you more confidence that they're likely to be pathogenic. And then you can do lots of functional studies within cells in the lab to have a look at the function and use a normal copy of a protein or a DNA sequence versus one with a variant that you think might be damaging, and compare how they behave in the cells to see whether there's a difference.
Okay, that's great. Thank you. So I actually want to touch a bit more on your lab experience, because I know as a senior lecturer, you're actually an exception because you're still in the lab as well. You still have got your touch with the lab work. So can you tell us or give us an example of a functional method that you yourself have used in the lab to identify the function of a variant that you might have found in your studies, in layman's terms?
Yes, possibly. So one of the things that I mentioned in the SmartB1 gene was this 3' UTR variant, so a variant after the end of the coding sequence of the RNA transcript, but still within the RNA transcript. And what that seems to be doing is to regulate the expression or the levels, the amount of RNA that's produced. So one experiment that I've done to look at that in the past is to create some of the construct with these specific sequences in a plasmid vector that can be put into some cells, and to have those vectors tagged with a fluorescent, like a green fluorescent protein, so that when you put them into the cell, you can see whether they're being expressed in the cell, because they will glow green, and you can look at that under the microscope. And if we look at the levels of RNA and of protein that's being produced, you can see how many of these cells, if you put a fluorescent protein tag onto a SmartB1 transcript that has a missense mutation, you can say, is it expressed more or less? And if you look at these variants in the 3' UTR, you can say, how are they functioning? If you look at a known function of SmartB1, which might be involved with, say, cyclin D1, something to do with cell regulation, is it still active? Is it still doing that? Can you look at levels of fluorescence, or can you look at levels of luminescence in terms of activity in the cell? If you put a wild-type version versus a mutant version of the transcript and then of the protein into the cells, does it change the output in terms of fluorescence or bioluminescence, and give you an indication of whether or not it's doing the same thing that it would normally do in the cell? I don't know if that's too layman's terms or not layman's terms enough.
I don't know. I'm sure if somebody asks a question, we will find out. So just to follow up on that, you mentioned about measuring RNA levels. So can you tell us what kind of methods, what are the names of these methods that can be used, for example, to measure the levels of RNA?
So a lot of them tend to be PCR based again, so you might amplify the region. You can use quantitative PCR, where so when I showed you the diagram of you have a region and you amplify it and you get lots and lots of copies, you can have a look at certain stages of the amplification process using a more sophisticated PCR machine, and have a look at when it's increasing in number, is it increasing and then stopping, or is it increasing and it keeps going? And depending on how many copies of a particular sequence are in your reaction, you might get more or less of a signal in the qPCR.
That's great, thank you. So thanks very much for that. And I think everyone would be interested, or would be I think it's very interesting that PCR is such a versatile method that you can just use it to look at RNA levels or DNA level or changes in DNA for a genetic study in cancer or any other disease. And at the same time, you can use it to test whether or not someone has got or infected with COVID. So I think it just shows how important or how fundamental this method is. So thanks very much for talking about that in your talk today. So the other thing that I wanted to ask is, you mentioned about microsatellites. And I know you have given some level of description as well. But can you tell us a bit more about microsatellites and why they might be significant in human disease?
Well, so the way that I was describing them in my talk was really to show what kind of variation you would find in these regions in normal people as well as people who have a particular disease. They tend to be used as markers so that you can say, in this particular region, there's some DNA sequence which is very repetitive. And during the DNA replication process, it's possible that you can end up, like the machinery that copies the DNA might make a mistake, it might slip, and it might add an extra repeat, or might chop one out by mistake just because it's repetitive and it's difficult for the machinery to tell when it's got to the end. So you get people who end up with different numbers of copies in these regions. And the microsatellite regions are areas where there's some known variation in the general population in these areas of repetition, and that you can sometimes use them to determine which copy of your gene is associated with this particular marker. And then you can use that to find out whether you're looking at the right strand of DNA when you're investigating another genetic change. So in the region that I showed you, I think it was a triplet repeat. And there are a certain number of copies which you might have inherited, say, 11 copies of that region from one parent and then 14 from another. So if you amplify that region of DNA, you will end up on an agarose gel, where I showed you the black gel with the white bands on it. You would end up with 2 bands rather than 1, because each copy is amplified in a different size. And you can use that in this case for determining heterozygosity. So if you know that in a normal cell in that person, they have 2 different sized copies of a particular region, but if you look in the DNA, one of those copies is gone, it gives you an indication that you've lost some genetic material, or that you might have lost one set of material, but the other one might have been duplicated, and it's only representing one version of that region. So it is a bit difficult to explain, but that it can give you an indication of whether you have one different kind of size or 2 different sizes. And that can be used to map regions and look at, is someone from the same family the same or different in that region? So if you know that something is tracking with a particular disease, it gives you an indication of whether or not that person might be affected, even if you don't know a specific gene change, because you know they've inherited that whole region. If you have several microsatellite regions along a chromosome, you can map how many of them are the same and how many of them are different in various family members. So you can look at relatedness and whether someone might have inherited one, what would be called a haplotype, versus another. I think in terms of when you say linking it to a particular disease, sometimes these expansion regions themselves can be associated with disease, like Huntington's disease, which I think, did Dave Gerard mention that possibly last week?
Or if he didn't, I can't remember.
Okay, but some of these regions can be associated with a particular gene and might have a particular effect on a gene. And then if you have a lot of copies and you end up getting more and more copies, it can regulate gene expression and have a damaging effect. So it can be associated with a specific disease phenotype. But in other cases, if they're occurring not within a gene, it can be used as a marker to indicate relatedness.
That's great. Thank you very much, Miriam. It has been a pleasure to have you. And it has been an hour and 10 minutes. We made you, we kept you here for an hour at least. So I'm just going to thank you, and we will be again in touch. To follow up your work and everything. But before I let you go, as PCR is so fundamental to your work, I was wondering if you could help us to find a valid animation for PCR so we can share it with our audience and make sure that they actually understand how it's done and can see it visually as well. So would you be able to help us with that? Lovely, that's great.
Yes.
Thanks very much, and enjoy the rest of your evening.